honored to present my work to you today, and thank you very much to the organizers. Today I'm going to tell you our modeling approach, which has been specifically optimized to study behavioral paradigms with slightly more complex temporal dynamics, like navigation. In navigation, I think there can be several cognitive steps. To begin with, the mouse needs to form a discrete navigation goal in its mind. Like for example, whether I want to go to the left or the right terminal of the corridor. Afterwards, it needs to form this continuous navigation trajectory, which maps from location to local motion. In other words, this will tell the mouse what should I do when I am at a given location. And eventually, the mouse just needs to move. And this navigation cognitive steps will need to be repeated again and again as the animal passes through the maze. And they also needs to be coordinated in a dynamical way to assure the robustness of the behavior. So the overall goal of this project is to study how in a mouse brain does it transform a navigation goal into a moment-by-moment locomotive output. As a very brief introduction, many cortical areas have been indicated to participate in navigation. For example, there involves primary visual cortex, the sensory areas, and associative areas like PDC and RSC, motor areas like M2. This is definitely not a complete list of all the possible brain areas involved, but these are the four brain areas we're going to focus on today. Again, the goal of this project is to study how do the functional ensembles across cortical areas interact to transform the navigation goal into the moment-by-moment locomotive outputs. In mouse work, we will typically start by recording the neural activity data as the animal is performing the task. And from these neural recordings, we'll be able to inform the neural representations, for example, which test variables the neuron should care about. But the problem is we're not able to know how the neural representations are generated. Alternatively, you can do optogenetics, but they can be very time-consuming, and spatial resolution can also be limited by the available tools. So in this project, we want to take a computational approach. The hope is we can have a generative model, and when it is very well constrained by animal data, we can use the model to help us to infer certain mechanisms of neural computation. So what are some of the typically used models in the field? On the one hand, they have these task-performing models or agents in the language of machine learning. These models will be trained to interact with the environment in closed loop and generate desired behavioral outputs that can complete the task. While these models are proved to be very powerful for task performing, as neuroscientists, you might worry that they do not use brain-like representations. Well, I mean, if they do use brain-like representations, then that is super interesting and we should study why. But the hope is, can we have some more explicit questions? On the other hand, we have the activity fitting models. These models are explicitly trained to fit to actual neural data. And the models involved here can be as simple as a linear regression or as complex as an artificial neural network. But as an exchange, the worry might be that these models do not use brain-like might not use the algorithms and computations necessary to achieve the task. And this concern can be especially true when we are studying behavioral paradigms where the environmental input animals receive actually depends on their behavioral output. So there's some closely approach. The hope in this project is we can somehow combine the benefits from both domains. We want to have these two-in-one models, which after training could on the one hand perform the navigation task, and on the other hand be able to generate the observed interactivity and behavior of the animals. So how do we do that? We designed these R models, and we basically wanted to do two things. First, these models should be able to interact with the environment in closed loop. This environment takes in velocity, use the velocity to integrate position and use position to determine sensory experience like visual cue. This is almost like how an animal will actually interact with the real-world environment. And then this visual cue and the other environmental observations will be fed to the model, and through some input weight, arrive at the core RN structure. And finally, the RN gives out some velocity outputs that feed back to the environment and close the loop. But besides that, we also constrain the model by real animal data. So there are two parts. First, we'll fit the activity of each unit in the RN to to the activity of a real neuron from the brain, which to say we will have the same number of units in iron as the number of neurons we can record from the brain in a session. And besides that, we also play the velocity outputs to the outputs of the actual animal. Another, I think maybe the most important aspect of the model is we will train the model to fit single trial data instead of trial average. So first, why do we want single trial training? This is because we think the trial variable structure can tell us something about neural representations and dynamics, and I'll talk more about this in a minute. And then how can we do single trial training? I would say this is especially difficult in those navigation tasks because animals tend to run with different paces in different trials. Sometimes they run faster, sometimes they run slower. So we need to accommodate the model to that. In short, we are using this closed loop training procedure. The idea is we will let the model to determine its own running pace autonomously by injecting this environment, but then during training, we'll encourage the model to match the pace of the target trial. I can talk about this later, if you're interested in the details, but basically we will apply this model to this navigation task performed by my collaborator, Charlotte Part. This is a maze-based navigation task with a fixed association between visual cue on the wall and the desired turning direction. The mouse needs to run forward to go to the end of the maze and then run laterally to turn to the desired reward location. And then Charlotte did mesoscopic calcium imaging of the neurons from the skull brain areas I mentioned at the very beginning. And then our goal is to study how these brain areas and neurons in brain coordinate. As I said, the model is supposed to be both task performing and data fitting. So here on the left, I'm showing you the model can achieve good performance in making correct choices, even in test trials that outperform training. And also on the right, the model successfully replicated neural activity from the real mouse brain. And then what's more interesting is what we get by fitting the individual trials. So the claim is that by fitting individual trials, we can capture the collaborative structure between neural activity and different variables, or in other words, the neural encoding profiles of those different variables. To illustrate the idea, here I'm showing you the activity of 3 example neurons as a function of the mouse forward position and lateral velocity in a maze. So all of these 3 neurons prefer left trials over right trials, but they like different things. The first neuron likes a certain forward position, the second neuron likes a certain lateral velocity, and the third neuron is only active when the mouse is running at a certain velocity at a certain position. So there's a conjunction structure. Then it is encouraging to see that the model can tell these different representation profiles apart. We can then quantify the level of neural representation by a generalized linear model. Here, each dot is a neuron, and x-axis is how much this neuron cares about one of the 3 variables in the data, and y-axis is how much the corresponding unit cares about the same variable. So it's good to see that there's a linear, there's a positive correlation. And by the way, if you can see the red mark that corresponds to sample neuron, I'll show you above. Then intuitively, if we only have tri-average data, we will not be able to tell apart these 3 representations because this 2D plan will collapse to a single line that corresponds to tri-average lateral velocity. And then quantitatively speaking, when we only train the model on tri-average data, we see the positive correlation is gone. This really highlights the importance of training the model on individual trial data, and this basically tells us that the trial-by-trial variability or the residual activity is not a bad thing, it's not useless, it's actually quite informative. So now we have a good model, what are we going to do with it? The short answer is we can use the model to study the functional activity between the units and how these neurons coordinate in a circuit or explainable system. The idea is I choose to tell you about the 3 representations because we think it could somehow correspond to the 3 cognitive steps I mentioned to you at the very beginning. To be more specific, those neurons that have a conjunction of position and choice could potentially tell the mouse where should it go, in discrete manner. So here each circle is a neuron. Density means the neuron's preferred forward position. And here you can see we have these 2 choice selective sequences that tell the axis of forward position. And at this given time, only that yellow neuron is active. And then if we integrate navigation trajectory as the mapping from location to locomotion, then that is exactly what this conjunction between position and velocity is doing. And eventually, velocity neurons are, of course, for movement outputs. And the activity of these neurons will be updated as the mouse proceeds forward through the maze. Now we want to use the model to give us some insight on the causality, whether and how these cell classes contribute to the task and how do they coordinate in the circuit. To do that, we start by doing this in silico projection inhibition screening. The idea is we want to inhibit the projection from certain source neurons to certain target neurons, and we want to see by inhibiting which projection do we get a very negative influence on the behavior, which will be marked by the red color. The overall idea is we think each projection in this table might correspond to certain pathways or circuit in the brain, although the mapping is not direct because there are so many unrecorded brain areas. But the idea is we want to recover some insight on a circuit mechanism underlying the navigation behavior. So here I'm showing you very preliminary data, so we are still optimizing the procedure, but I hope you can see that at least not random, there are some patterns. And then for the sake of time, I'd like to draw attention to the single row and column that involve the position velocity conjunctive neurons in RC. So we think these neurons are interesting for three reasons. First, if we inhibit the interactions between them, we get a strong negative influence. This means these neurons, they recurrently connected to each other and they're doing something. And secondly, we see there are strong reciprocal interactions between this position velocity neurons in RC with the velocity neurons in PVC and M2. And PVC and M2 are more motor-like areas, so there are some readouts from those populations. And finally, since we have this RN model, we can actually look at the connectivity matrix of the RN to see what are these position velocity neurons in RC actually doing. So they actually find that these neurons are connected in a like-to-like manner, which means these neurons will excite each other more if the pre and post pairs have a similar preference of position and velocity. So this like-to-like pattern might remind you of something of fixed point attraction, and that is exactly what we find. So here we do some eigenvalue analysis. Here I'm showing each dot is an eigenvalue and the real part and the imaginary part. So the idea is we find that the real part of all the eigenvalues of this sub-connectivity matrix fall below 1, which means there are some attracting dynamics going on. And we also did some other simulations, and we find that the attractor is actually contact dependent. And here contact basically means whether the mouse is in a right or in a left channel. I want to remind you that these position velocity neurons are position dependent. So what we think is happening globally is we might have many of such contact dependent attractors, each preferring a certain forward position. And many of these attractors can be chained along the forward position axis, which eventually define the navigation trajectory. And we also did a bunch of other circuit analysis. I'll just go through them very quickly to give you a global view. We find that these velocity neurons in RC will receive contextual input from the choice-specific sequence. And at the same time, the velocity neurons will read out the velocity in a selective way. I'm happy to talk more about the dynamic analysis and the circuit analysis later if you're interested, but I'm going to wrap up now with this summarizing slides. By using this data constraint closed-loop single-child R model, we are able to do a bunch of analysis with it, and this gives us this hypothesis of the attracting dynamics underlying navigation, and most importantly, we can identify which circuit made up from which ensemble, the RC position velocity neurons that analyze the attracting dynamics. And we hope that this can inspire some, test the hypothesis that we can actually test it in most experiments. I'd like to end by thanking my advisors, Chris Harvey and Tim Rajan. They're amazing mentors, and I'm super lucky to be working with them. I'd also like to acknowledge my collaborators, Ryan Batman and Charlotte Out, and all my dear lab members in the Harvey Lab and the Rajan Lab.